Continuiamo la serata e allora chiederei a questo punto a Craig Mello di salire sul palco, il nostro ospite gradito. Please, Craig. Chiederei a Nicola e ad Agnese di... This is your place, wherever you want to sit. No, that's yours. Yes, you are in the center, of course. That's mine. So, scusami. Scusa, è vero. Prego Maria Elena, a te. Io fatti sedere intanto, scusami, non prendertela con me adesso.
Buonasera, io sono Maria Elena e faccio parte di WeScience, il gruppo giovani di BergamoScienza. Prima di iniziare vi chiedo un momento di attenzione e vi ricordo che collegandosi al sito bergamoscienza.it e cliccando sul pulsante live è possibile postare su Facebook la diretta dell'evento, inviare domande ai relatori o invitare i propri follower a seguire la conferenza in streaming. Le domande dal web vengono ricevute da noi volontari che ne proporremo una selezione durante il dibattito in sala. Le conferenze della 22esima edizione di BergamoScienza sono rese accessibili a persone con disabilità motorie e sensoriali, cercando di garantire luoghi privi di barriere architettonica e mediante servizi di traduzione LIS. Tutte le conferenze sono videoregistrate e alcune saranno trasmesse anche in diretta streaming. Le conferenze con ospiti stranieri, come stasera, prevedono una traduzione simultanea. Le registrazioni verranno pubblicate sui nostri canali ufficiali a termine del festival. Questi servizi sono resi disponibili grazie a Fondazione della Comunità Bergamasca e Fondazione Banca Popolare di Bergamo, che insieme a BergamoScienza sono impegnate a promuovere inclusione e accessibilità. Per la traduzione simultanea è necessario scannerizzare il QR code che vedete proiettato alle mie spalle. Il QR code vi indirizzerà direttamente alla Converso Web App, dove potrete selezionare la lingua dal menu a tendina. Per l'ascolto è necessario una coppia di auricolari, vi chiediamo di usare le vostre, ma per chi ne fosse sprovvisto potete chiedere ai volontari in sala. Vi do quindi il benvenuto all'evento di questa sera, che è intitolato RNAi Interference, la scintilla molecolare delle informazioni. Avremo il piacere di ospitare come relatore Craig Mello, premio Nobel per la medicina nel 2006, che dialogherà con Gianvito Martino, Agnese Collino e Nicola Quadri dell'Associazione BergamoScienza. Vi preghiamo di silenziare i vostri apparecchi elettronici e di partecipare numerosi alle iniziative di BergamoScienza. Grazie per l'ascolto.
Grazie. Allora, adesso vi abbiamo riservato una sorpresa che non abbiamo voluto divulgare perché sennò era abbastanza complicato. È la sorpresa che abbiamo la presenza del nostro grande mister di Giampiero Gasperini, che ha voluto venire a trovarci. Grazie, grazie molte. Allora, perché siamo qua? Perché siamo qua? Allora, siamo qua per questa ragione. Nel 2018 Craig venne per la prima volta a BergamoScienza e scoprì che a Bergamo c'era una squadra che si chiamava Atalanta. Lui aveva appena fondato una biotech che si chiama Atalanta Therapeutics nel 2018, che è una biotech. Parlavamo prima con Craig che per darvi la dimensione, nel secondo round di finanziamenti dell'anno scorso ha raccolto più di 100 milioni di dollari ed è una compagnia, una biotech che grazie alle sue scoperte usa le sue scoperte per creare farmaci per malattie neurodegenerative e in generale per malattie del sistema nervoso. Sapete che nel mondo una persona su 4 soffre di una malattia neurologica o psichiatrica. Quindi è una vera piaga in questo momento. Craig, poi ne parleremo, con questi piccoli RNA, questa scintilla di RNA, riesce a entrare in maniera chirurgica nelle cellule del cervello, del midollo e così via, e far cambiare a loro il modo di produrre le proteine perché interferisce accendendo o spegnendo dei geni che possono essere malsani o alterati e causare la malattia. Nel 2018 il nostro mister, ovviamente è arrivato a Bergamo nel 2016, ma per chi ha una fede atalantina incrollabile come me, la devo confessare, nel 2018-19 fu l'anno trionfale per l'Atalanta, perché il nostro mister fu in grado non solo di farci arrivare terzi in campionato, ma di arrivare per la prima volta in Champions League. Cosa per uno come me che segue l'Atalanta dal 1971 non era qualcosa di particolarmente frequente, diciamo. E quindi ci è piaciuta, diciamo così, ci è piaciuto questo gemellaggio, perché Craig poi è venuto a trovarci, anche 2 anni dopo, quest'anno è tornato, ma poi ne parleremo, e addirittura quest'anno si è presentato col cappellino dell'Atalanta. Oltretutto... e mi dicono, ma non so se è vero, che addirittura quando scoprì dell'Atalanta comprò le magliette per l'Atalanta a tutti i dipendenti di Atalanta Therapeutics, this is true, e appunto lì gliela feci indossare perché in qualche modo... Quindi l'Atalanta è anche nel cuore di Craig, non solo nel nostro, e quindi il tributo al nostro mister ne viene da sé. E quindi abbiamo pensato di fare un regalo a Craig, però non se la cava perché qualcosa le faccio dire, quindi inizialmente, intanto... E questa è la magna. You see, this play, no, this part of the, this is the most important part of the story, because we won. I say we, I mean I'm not part of it because I don't play, but anyhow I feel part of the story. The UEFA Europa League in 2024, che è stato was the major achievement of our society. So, let me just, dobbiamo lasciare il nostro mister perché, ahimè, domani deve andare a me, o per fortuna a Bologna, dove faremo il nostro dovere, mister? Diciamola così. Ma volevo in primis ringraziare il nostro amico comune Ivan che ci ha dato la possibilità di ospitare Gasperini. Grazie Ivan, è il nostro ristoratore, diciamo, preferito, sia mio che del mister. Però ne parlavamo prima, una domanda sola. Dicono che lei, insomma, pretenda molto dai giocatori in termini, diciamo, sia fisici, però prima mi ha detto che anche dal punto di vista mentale è fondamentale l'equilibrio che un giocatore deve avere per diventare un grande campione. Ha voglia su questa cosa di rielaborare un pochino quello che mi ha detto, cioè il suo pensiero rispetto al fisico versus la mente, diciamo.
No, ma il mio pensiero, l'ho ribadito tante volte, molto spesso negli atleti, in particolare nei calciatori, vengono valutate le capacità tecniche, le capacità atletiche, fisiche, molto poche volte in quello che è lo scouting, lo screening del giocatore, si valutano magari le capacità cognitive, le capacità di essere competitivi, di essere reattivi e tutte quelle componenti che fanno parte un po' della, io dico della personalità, ma che poi riguardano quell'emisfero abbastanza sconosciuto che per fortuna con persone come noi riusciamo sempre ad avere qualche chiarezza in più e che invece per me sono fondamentali nella valutazione di un atleta, di un calciatore, di una persona. È chiaro che bisogna avere delle qualità a prescindere, ma nella mia esperienza chi aveva queste doti ha sempre, anche magari con dei mezzi tecnici inferiori, ho visto tanti talenti importanti dal punto di vista tecnico non riuscire a raggiungere traguardi pensati. Io ho visto tante persone, tanti giocatori con mezzi fisici, atletici magari meno importanti, ma con una grande qualità morali, grande qualità di cervello, diciamo così, che hanno fatto delle carriere straordinarie e sono stati degli esempi anche per il nostro sport.
Ecco, l'ultima curiosità è come valutate voi l'aspetto cognitivo rispetto a quello fisico? Abbastanza semplice, diciamo. Cioè avete dei team dedicati, avete persone che si occupano di fare testistica psicologica piuttosto che... Cioè, oppure è la sua impressione che ha rispetto al comportamento del
paradossalmente devo dire, per quanto nel nostro ambiente insomma ci siano dall'aspetto economico delle cifre molto importanti, difficilmente ci sono questo tipo di valutazioni, anzi direi quasi mai, meno quando poi ti trovi a confrontarti poi direttamente a cose già acquisite.
Cioè se ne accorge lei di come si
Accorgi
comporta il
poi,
giocatore.
sì, puoi prendere notizie, puoi prendere questo sì, ma questo si allena bene, questo è un bravo ragazzo, questo è una persona educata, non ha particolari comportamenti sbagliati. Sì, queste notizie vengono prese in questo senso, sì, però poi dopo sia nei giocatori già affermati ma anche nei giovani, ad esempio, no? Quando vengono fatte delle valutazioni anche su base di nuovi talenti, difficilmente si guarda questo aspetto. Anzi, devo dire che molto spesso nel calcio, e questo io lo ritengo un grande limite, già da ragazzini quando arrivano a 16-17 anni vedono che hanno delle possibilità di giocare nel calcio, prima cosa smettono di andare a scuola. E questo è un grande limite, perché questo va sempre molto allenato e è un allenamento anche per noi, poi voi lo sapete sicuramente meglio, ed è importante avere queste capacità, ti permettono comunque di migliorarti, di crescere più che allenare il quadricipite.
Grazie mille mister, è stato gentilissimo.
È un piacere, grazie davvero. Grazie.
Grande piacere averla qua, grazie ancora veramente perché ci ha fatto molto piacere. Allora due, certo. Craig, if you wanna wear, I mean, just show the t-shirt for, and if you just take out this, sorry, for a few pictures. Sorry, Craig, but this is what we need.
Provo a fare numero 1,
ma quello è il portiere, non si può. Grazie ancora, grazie molto, veramente grazie. Spero di rivederla e buona trasferta a Bologna. E sappia che c'è nel cuore tutta la città. Grazie. Va bene, so we can start talking about life. Allora, I have to talk in Italian, I think. Bene, il tema di stasera è un tema molto interessante. Nicola ed Agnese mi daranno una mano, ovviamente. Craig ha chiesto di fare una conversazione più che una lettura, diciamo, magistrale classica. Quindi mi ha chiesto anche di esortare il pubblico presente a partecipare. Ovviamente siete tutti... siamo tutti contenti di poter ascoltare anche il vostro parere. Vi chiederei solo, quando apriremo la discussione, faremo un'iniziale introduzione, di fare domande possibilmente brevi, perché sennò diventa complicato poi seguire l'andamento del tutto. Quindi sostanzialmente partiremo con un'introduzione di Craig, che ci dirà dove siamo, dove stiamo andando e che cosa conosciamo della vita. Io posso solo dire che dal punto di vista biologico è uno degli argomenti in assoluto più affascinanti, come potete immaginare. Vi posso dire che non sappiamo tanto di come sia originata la vita, lo immaginiamo, ma il limite più grosso è che non riusciamo sperimentalmente a ricostruire le condizioni che c'erano 3 miliardi e mezzo di anni fa, quando la prima cellula Luca è comparsa sulla Terra. Però diciamo molta della ricerca, anche perché semplicemente sembra che il tutto sia nato da una meteorite gigante che è caduta sulla Terra, e quindi è ovvio che in un laboratorio riprodurre una meteorite che cade sulla Terra è un po' complicato, come potete immaginare, quindi è difficile proprio tecnicamente, ma a prescindere da ciò i biologi molecolari come Craig certamente hanno iniziato a capire quali erano le caratteristiche della cellula così come la conosciamo noi e come una struttura così complessa potesse in qualche modo autoformarsi. Perché Craig è stato fondamentale in questo percorso? Perché la sua scoperta ovviamente introduce una categoria di molecole, di macchine molecolari, che durante l'evoluzione sono state fondamentali. Perché si dice in particolare che la prima struttura cellulare fosse un ribosima, si dice, quindi una sorta di struttura che conteneva dell'acido ribonucleico, dell'RNA, ma nello stesso tempo era in grado anche di fungere da enzima. Tra l'altro gli enzimi di cui domani sera parleremo con Frances Arnold, i suoi di sintesi, però che riproducono quelli in natura. Quindi, insomma, ci sono esperienze, le abbiamo viste, se vi ricordate Jack Sosta, che è stato qua con noi, ci ha parlato di queste piccole membrane autosigillanti, per cui la membrana cellulare, tutto sommato, che è veramente rilevante, si può costruire in parte, ma insomma, detto ciò, il percorso è ancora lungo e molto complicato. Concluderei qua, lascerei la parola a Craig, che inizia a introdurci all'argomento, e poi iniziamo a chiacchierare col pubblico. Thank you very much for being here, Craig. It's a real honor and pleasure for me. Thanks. Prego.
Hello, everyone. It's great to be here. And if I keep coming back, I'm going to have to learn Italian. I apologize that I can't speak your language, but I'm 1/4 Italian. My grandmother, Primiano, is from the Naples area, Campobasso. So it really is wonderful to be here. You are so lucky. You live in one of the most beautiful places I have ever been. This is just gorgeous here. I came to Italy to speak in Pisa, where they were celebrating 3,000 liver transplantations there in the hospital in Pisa. And it was such an incredible, emotional, and wonderful experience to see those wonderful doctors, you know, actually moved to tears by their own patients, you know, and some of the things their patients said, you know, and the gratefulness of their patients. It's really wonderful to see people with compassion for each other. You know, we read in the news too much about hatred and division between people, but we share so much. You know, we have such a wonderful, incredible history together on this really fragile and beautiful planet. I really love science because the more we learn about the world, the more mysterious the world becomes. It's truly, truly incredible, the planet that we live on and the living things that are around us. When I was a child waiting for school one day, I remember reading in the newspaper while I was eating my cereal that the human gene for insulin had been cloned and put into bacteria, and bacteria could read the human genetic code and they could make insulin. And the article was about how powerful that would be, how wonderful, how transformative, how great for medicine. But not once in that whole newspaper article was there an answer to the question that was burning in my mind. How can a bacterial cell read the human genetic code? How is that possible? Why is that possible? And that began my career. For me, that day, I decided I wanted to become a biologist. I wanted to learn how organisms handle information. And it's truly, you know, Gianvito mentioned that we can't reconstruct the beginning of life, but we live in such a fascinating age because we now can read the information of life. We can read what's called the DNA sequence, so you've all heard about it. And DNA is where information is stored. It's like a library where the books are, right? That's the information that makes us work. But there's another form of information in living things as well. And that information is called RNA. It's like DNA. It's very much like DNA. But it's the spoken language of the cell. And many of you probably, even if you're not a biologist, heard the word RNA a lot during the pandemic and over the last few years, right? RNA, mRNA, vaccine. You know, most of you probably don't know what an mRNA is. It's jargon, but it's like it's basically a sentence or a paragraph or perhaps a chapter of a book, and it's being spoken by living things. So the pandemic began in China, and this little RNA virus that came from a bat jumped into a human, infected a human somewhere, whether it was in a laboratory there or in one of their food markets. We don't really know. Their people argue about it back and forth. But that virus, within 2 or 3 years, infected probably every single one of you in this room. It swept the entire planet. The information from that virus infected you. It made you sick. It copied itself inside of you. That is incredible. This virus, 30,000 letters of code, just 30,000 letters of code. That's a really, really short book. It can copy itself, replicate, spread, and our bodies had to deal with something new and develop immunity and, you know, recover as a society from this devastating virus. But it's just, this is common in biology. This is why I call, you know, that maybe, I don't know if you saw the title I had chosen, I called RNA interference this mechanism that we discovered. Andrew Fire and I shared the Nobel Prize for discovering a mechanism called RNA interference. It's an RNA mechanism that blocks information. And I called my title A Molecular Spark in an Information Inferno, because I'm going to stop. I don't want to lecture you too much. I want to be able to take questions, because I have found that to understand each other, because of the barrier, the language barrier between science and the layperson and the general people in the audience, you might not understand some of the words, but I want to try to communicate with you. The answer to my question when I was reading the newspaper, why is it that the human gene can be read by the bacterial cell? The answer is that information is constantly flowing horizontally across from person to person, from microbes to humans. Their information is flowing. It's moving around. It's infecting us. It's changing us. It's helping us. It's killing us. Information is both powerful and, as we all know, dangerous, very dangerous. And so a lot of our biology has a role in protecting us from information, but also capturing information. So over the history of life, it's now completely clear from modern biology, it's completely clear that you do not only get your information from your mother and your father. Information has flowed into and across species from our environment. You got infected, that's information. This kind of information is incredibly important in biology, and now we can begin to use information to make drugs. This is why we formed this company, Atalanta Therapeutics, to try to make therapies based on information. We can give a patient a piece of information that will regulate a gene in their brain. This is incredible. I mean, I did not even imagine this back when I was reading that newspaper at my kitchen table, but we can do that now. We can make drugs and therapy with information. And that's why many of you who are sick will have your doctor may ask you if you'd like to have genetics done on your cancer or your family to learn to help understand why are you sick. And so I want to communicate with you about your information, about biology and about how information works, because it's truly fascinating and also extremely powerful. And there's a lot of fear that we have in our society over information, over biological information. What is a vaccine? What does it do? What is a genetically modified food? Is it dangerous? These are the kinds of things I would love to hear your questions. I'd love to talk to you more about the exciting opportunities to now create new medicines based on genetics. And I'll stop there for now.
Grazie Craig. Quindi, se siete d'accordo, inizierei a chiedere a Nicola e Agnese di iniziare con le domande. Intanto chiunque abbia intenzione di farne si prepara, poi i nostri ragazzi sono già pronti con i microfoni, basta alzare la mano e iniziamo questa conversazione. Sostanzialmente Craig è aperto, diciamo, a qualsiasi tipo di domanda ovviamente legata alla vita e a come questa informazione legata al DNA e all'RNA poi determina quello che è tutto quello che noi vediamo di animato, diciamo, attorno a noi. Quindi è un pochino questo il ragionamento. C'è qualcosa di questo che non vi è chiaro, che volete chiarire, che volete capire? Ha parlato di organismi geneticamente modificati, ha parlato di una serie di temi molto caldi. Ecco, questa è l'occasione per chiarirli. Agnese.
Io grazie mille davvero per questa panoramica e per l'opportunità di poterti fare delle domande. Io volevo partire con una considerazione personale per farti una domanda personale. Cioè, io qualche anno fa facevo ricerca in ambito oncologico e è emozionante pensare che...
Aspetta un attimo, Agnese. Potete sistemare il...
No. Yes, okay.
È partito, si sente. No problem. So, dicevo, partirei con una considerazione personale per poi farti una domanda personale. Ecco, io fino a qualche anno fa ho fatto ricerca in ambito oncologico ed è emozionante qui oggi essere seduti vicino alla persona che ha scoperto un meccanismo che io stessa ho usato in laboratorio nei miei esperimenti. Quindi, insomma, è sempre bello vedere come anche le cose che si fanno oggi in laboratorio non sono scontate. Qualcuno ci è arrivato prima, le ha indovinate in qualche maniera, le ha scoperte o le ha messe a punto, e insomma queste persone sono qui e lo possono raccontare, e anche quella è parte della fascinazione della ricerca, non solo che cosa si scopre, il risultato, ma anche il come ci si è arrivati. La domanda che volevo farti invece, Craig, è questa. Tu sei figlio di un paleontologo. Come ci si sente da figlio di paleontologo ad aver scoperto una parte di questi meccanismi di informazione così antica di fatto?
Thank you. And also the son of an artist. My mom is an artist. And my father worked in the Smithsonian, which was a beautiful museum in Washington, D.C. I think this is partly why I was so surprised by that newspaper article. To read that the human and the bacterial cells spoke the same language surprised me. And I didn't see any surprise in the scientific community. I thought it was odd. How come they're not surprised? You know, it's like you're reading like, where are they going to say this is surprising? And there's nowhere in the article it said this is surprising. It's surprising. It's amazing because it's billions of years. That is really, really long. The time between the dinosaurs and today is 60 million. That's a small number compared to billions of years. Living things are so ancient And we know a lot about them now because everything alive, we can read their genetic code. We've done it. You know, the genetic code inside of us is what my dad would have called a living fossil. It's a living fossil because the genetics that makes us work made the dinosaurs work. It made something called trilobites work. It made the organisms below the Cambrian 540 million years ago. It made them work. And they were already just as sophisticated as we are. They were every bit as capable as we are. They had all the tools. They had the namesake Atalanta Therapeutics is named after the Argonaut, who was a woman on Jason's team. She was very, very fast, but the namesake in biology is a search engine. It's actually just like Google. It takes a short piece of genetic code and it uses the information in that short piece of RNA to find matching information, just like you do when you type in something into your phone to find a restaurant. You are doing guided search. Organisms invented that billions of years ago. They already had it. And some of you, who in the room has heard of CRISPR?
CRISPR-Cas9.
CRISPR.
Sulla mano, non siate...
Yeah, okay. That's good. CRISPR is guided search where the search, it finds DNA and then it precisely cuts the DNA. Oh, it can actually now we know there's many, many types of CRISPR too, but the one that is most famous allows us and bacteria to use guided search to find and precisely cut DNA where the guide says. You know, the guide is the information, all that you have to give to the enzyme, which is made out of protein. And I always love, you know, because you guys, a lot of you don't know what a protein really is. It's just, you know, it's not a white powder that you put in your smoothie in the morning. Proteins they're encoded by the genetic information, which is one of the innovations of life that's very ancient. And it's why, you know, when we talk about insulin, we're talking about a protein, okay? So to assemble the insulin protein, you have to read the genetic code, and you read it 3 letters at a time. And there's a special, you know, translation system that reads those letters that are in this chain. Okay, now we're talking about mRNA, okay? So there's an mRNA for insulin, okay? Just like there's an mRNA vaccine. There's a gene in the DNA, gets copied into RNA, and then a translation system reads the genetic information. It reads it 3 letters of code at a time, and it moves along the code. Every time it reads a certain 3 letters, like A, T, G, there's 4 letters in the alphabet, A, T, C, and G, okay? That's it, just 4 letters in the whole alphabet. It's not that complicated, but they spell about 20 different words in terms of amino acids. These are building blocks of proteins. And everyone really should know this. It's not that complicated. It's much simpler than it would be for me to learn Italian, right? I mean, much simpler. But anyway, proteins are chains that are built by reading the genetic code. So if I want to make insulin and I'm a bacterial cell, all I have to do is read the genetic code and assemble the amino acids into a chain, and that you get from an RNA polymer, you make a protein polymer. Now you have a protein that folds up, and they can fold up beautifully into three-dimensional shapes that can carry out functions for your body and for the cell. And it's truly beautiful. Though I can't do it justice, but just imagine how living things work. They are informational molecules that can do chemistry. They can program the incorporation of building blocks to build these elaborate structures that carry out functions for the body. And so bacteria could read the human message, the human messenger RNA, and build a protein. And when that virus swept around the world, it brought with it an RNA that encoded viral proteins. So when it infected you, that virus would be able to have its RNA translated into proteins that make up the coat of the virus, that allow the virus to, get encapsulated into these little particles that can then spread. When you breathe, they would float in the air, and they would go from person to person inside of a little protein shell. Beautiful structure that can hold a little piece of genetic information, and that could float right through the air and infect a bunch of you in this room. Hopefully it's not happening right now. But living things are incredible, and they're ancient. And so I still talk to my dad about it. I love your question. But I actually find my mom also to be quite inspiring, because she often will point something out and say, Craig, what's that? And usually I say, Mom, I don't know. And she'll say, well, you should work on that. And she's right, because what's beautiful is often what's interesting, and what's interesting is often important. So I learned, I really lucked out to have the parents that I have. I learned so much from their perspectives, and I feel like, I still am learning, and this is partly why I really love to do the event here. You guys, this is amazing what you do. Bergamo is the capital of science, Scienza, in the world, in my opinion. You guys are great. But I definitely would love to hear more of your questions. So.
Thank you very much, Craig. You're very kind. Nicola.
Dai, faccio una domanda, poi vediamo se qualcuno è curioso e vuole farne una. La mia domanda era, hai parlato molto di informazione e di come le cellule usano le informazioni per costruire delle macchine, svolgere delle funzioni. Credi che sia diverso, che una cellula sia diversa da un computer? Credi che ci siano comunque delle differenze, o vedi le due cose come molto simili? Dovremmo pensare forse che in futuro la biologia e diciamo, i meccanismi che i biologi molecolari come te stanno scoprendo verranno utilizzati e incontreranno un po' l'ambito della robotica, l'ambito della computer science, e se sì, quando accadrà?
That's a great question. It's interesting because we are on the cusp of something new, this artificial intelligence thing. And I know your theme is intelligence here in Bergamo this time. But I think we have a lot to learn about how organisms handle information. First of all, information, as everyone knows, is very dangerous, right? You need to test the information carefully before you use it, because you could get into lots of trouble. Mostly you could lose all your money, I guess, is the big problem. But no, information is, it's incredibly important, and I think what's really remarkable is how living things can propagate information so flawlessly. And we still don't really understand how they do it. I think we can learn from them, though, if we can understand it better, because they have every generation the information gets copied, and our body intentionally, seemingly, shuffles the information around. So everyone in the room is a recombinant. You are all made by recombining the chromosomes from your grandparents. Actually, your parents shuffle their DNA during a process that makes the egg and the sperm. That DNA gets broken up into little bits and then rejoined to make new chromosomes. That's how you get a mixture of the genes from your grandparents. They get, you know, swapped around. So your chromosomes are all different from your parents' chromosomes. They've gotten altered during the process that made the egg. It's fascinating. So I think there's really something interesting else that's going on, because as you probably all know, our DNA is made up of lots of remnants of viruses. So, you know, everyone's heard of human immunodeficiency virus, HIV. HIV is a very common type of virus. Our DNA and every organism, almost every cell, even plants and fungi, have viruses just like HIV. They are 17, 18, like 20% of our DNA is from those viruses. That's all. It's like our genome, our DNA is full of stuff, they call it junk, but it's not really clear whether it's really junk. You know, it's like, it's there and we're not sure why. I think the reason it's there is because the information, like I said, is flowing horizontally. So these are remnants of past infections that jumped into humans many millions of years ago, and they are still moving and changing us. So we're getting shuffled. The deck is getting shuffled, right? And so if you're making a new artificial intelligence, maybe we could learn from that. Maybe we need some way that our computers will write code but make mistakes. You know, like set it up on purpose so that it creates mistakes. I mean, this could be bad, right? I mean, I'm not sure this is going to go in the right direction, but I think it's a fascinating, you know, in nature, the survivors have to have a working program, right? Because if the next generation, if you're a lion and you don't have teeth, you know, you better have some other trick, right? Nature's going to select. That's why Darwin called it natural selection, because nature selects the fittest. Who's going to select the fittest computer program? I don't know, but it could be a bad situation for humans. I'm not so worried, because I think humans, we need all the help we can get, and it's not clear that we're doing such a good job by ourselves. So. But I'm happy. It's an interesting. I enjoy reading about artificial intelligence and you know, all the things that can go wrong. But I hope some things go right.
Bene, possiamo iniziare a aprire le domande. Ovviamente non potevi che essere tu il primo a alzare la mano, perché oramai questa è una tradizione. Domande brevi. Forza ragazzi, con i microfoni, ce n'è lì una. E poi ce n'è una anche là in fondo, così ci prepariamo. Alza la mano, scusami che ti vediamo. Guardate là ragazzi, guardate là, state attenti, andate là. Ok, vai, devi accenderlo. No, non così, c'è il tasto sotto, dategli una mano. Vai.
Grazie per questa opportunità. Io sono ignorantissimo della materia e conosco molto poco anche l'inglese. Dunque, se dico una cosa di questo tipo, all'inizio c'erano gli organismi unicellulari, poi ad un certo punto, non so quando, c'è stato il maschile e il femminile. In ogni cellula ci sono 22 coppie di cromosomi, credo.
Cellule umane?
Sì, cellule umane che sono nel femminile
23.
2XX e invece nel maschile sono 21 XX e 1 XY. Questo mi indurrebbe a pensare che praticamente il maschile è derivato dal femminile, è un'evoluzione del femminile. Questo contrariamente a quanto ci hanno insegnato tradizionalmente nella religione, che la donna è nata da una costola dell'uomo con tutte le conseguenze.
Speriamo di aver superato questo.
Effettivamente il maschile deriva dal femminile oppure no? Grazie.
Did you get it? The male comes from the female and vice versa.
Great. The human has sexes that are this way, but there are many, many other ways that organisms share information. In the human, we have a special chromosome, as you mentioned, called the sex chromosome. The males have a Y chromosome. You know, interestingly, there's another word for that in a really interesting fact that I found really intriguing. It's called a distributed genome. So that means that the genome is not carried by one individual. Women require men. That's one thing men are good for. We bring the Y chromosome. Without it, you don't have humans, okay? So it's an interesting thing, though, because lots of organisms mate. They exchange information, you know, to diversify, to share differences that are, you know, important for survival. You know, and bacteria do this too. Bacteria have a vast distributed genome. They don't just have an X and a Y. Bacteria have these little things, little chromosomes that are often called you know, episomes, because they're not stuck into the main part of the DNA. They're just floating around, and bacteria mate. They inject each other. They like set up an apparatus that pokes a hole in a neighboring cell, and they will send information into the other cell. And this is why in the hospital, resistance to antibiotics spreads so fast, because bacteria are mating constantly. So which came first, male, female? Neither. Information flow came first. Organisms were sharing information for billions of years. Sex, mating, that's just a way of sharing information. So other organisms have other solutions. Bacteria, did you know that bacteria have a bigger genome than the human? Most bacterial populations have a vastly distributed genome. They have 80,000, more than 80,000 genes. We have maybe 20,000 in the human genome. And this is the other sort of, my talk is, part of the title is a molecular spark in an information inferno. Living things are living inside of a inferno of information. This is why that virus caused so much trouble. It swept around the world. In the microbial world, they live inside this fire. They really do. In our genome, we brought the fire inside. We sort of have a candle burning. We have our, you know, genetic elements are jumping around and moving and inside of our DNA, and our genome is huge, and it's full of, you know, the stuff we don't, like the viruses I told you about that transpose, we call them transposons because they're pieces of DNA that jump around. But bacteria, even from different species, bacteria can share information very easily, and they constantly do. And they, and that's not even talking about viruses, which are, for every bacterium, there's thousands of bacterial viruses. And so, yes, everything in the whole world is sharing information. And sex, chromosomes, they're just another example of information that's shared. It's distributed over the population. And there's, this is true in especially in the microbial world, where living things share information at an even more incredible rate than we do as animals.
Dunque, c'è una domanda in fondo. Io qua voglio aggiungere, poi arrivo anche da lei in seconda fila, che il sesso in biologia è semplicemente uno strumento riproduttivo, non c'è nessuna sovrapposizione, diciamo, culturale o ideologica rispetto alla femmina o al maschio. Cioè, questo deve essere chiaro. Gli aspetti che tendono a dividere il mondo in maschi e femmine sono aspetti puramente culturali, la biologia non fa questa differenza. E diciamo le cellule sessuali sono comparse quando gli organismi, appunto come dicevi tu Dante, erano pluricellulari, e in qualche modo nella riproduzione di questi organismi complessi necessitava di un sistema di selezione, che è quello a cui faceva riferimento Craig. E diciamo l'accoppiamento sessuale è dal punto di vista, diciamo, biologico il sistema più semplice, o perlomeno quello che finora consideriamo il più semplice, però la nostra specie si è riprodotta asessualmente, gli organismi viventi asessualmente per 500, mezzo miliardo di anni. Cioè, nel senso, siamo noi che abbiamo costruito poi sugli aspetti, ne parleremo con la Diane Rosenfeld, quando parleremo dei bonobo, e parleremo dell'antropologia che sta dietro in qualche modo la violenza sulle donne, o queste logiche di patriarcato, insomma, che iniziano a essere abbastanza, diciamo, poco tollerabili, perlomeno dal mio punto di vista, perché, insomma, il problema è serio. Però siamo noi che facciamo le differenze, non è la biologia che fa la differenza, la biologia è molto democratica, siamo noi che non siamo democratici nel condividere la realtà, ahimè, e dare etichette a persone semplicemente perché hanno, insomma, interessi o diversità rispetto a noi, ma questo è un problema complicato che però tratteremo, e quando viene Dan Rosenfeld vi consiglio di venire ad ascoltarla, perché lei è il capo del Violence Department di Harvard, è un avvocato importante che è anche un'antropologa di formazione, che ci racconterà delle storie molto interessanti rispetto a questo tema. Sorry Craig if I follow up a little bit what you said. Bene, c'era una domanda là in fondo e poi una domanda qua in seconda fila. Prego.
Buonasera. I suoi studi sul silenziamento genico, se non sbaglio, sono di fine anni 90. 2 anni fa è venuto qui il 14 ottobre ed era stato lanciato da poco sul mercato italiano in Clisiran. Ancora oggi però...
Scusi, può spiegare che cos'è, perché non tutti lo sanno.
È un farmaco per le dislipidemie, è un SIRNA, è un silenziatore di RNA. E, diciamo che c'è ancora molta diffidenza su questi farmaci, un po' di paura, anche se gli effetti collaterali, essendo farmaci talmente specifici, sono praticamente nulli. Però c'è ancora molta diffidenza, scusate. Perché secondo lei?
Grazie.
Well, the question really is about why people fear using a drug. It's also, in this case, you know, I think it's very, very natural that people fear what they don't understand. You know, and that's why I feel like speaking to you is important. You know, I value this opportunity, and I really thank you all for being here, because we all have a role to play. Because even though, you know, there's problems with democracy, you really need to have, it's like the best form of government. you know, that anyone's come up with. You know, unfortunately, it's not far away from perfect, but we all have to participate. And in order to participate, you need to understand what the issues are. So when it comes to something like a vaccine and you don't know how vaccines work, then you need to educate yourself, you know, so that you can make an intelligent vote that's good for society, a good decision for yourself and for your loved ones. And you need to do that. That's your responsibility as a human being. So the drug that was mentioned called inclisiran is now being marketed by Novartis as Leqvio. The company that made the drug was acquired by Novartis for $9 billion for just that one drug. And what that drug does is it replaces a drug that many of you are taking right now called statins that lower cholesterol. And instead of taking a pill every day that has lots of other side effects, you can take a single subcutaneous injection. It's like an insulin injection. For those of you who have diabetes, you're perhaps familiar, you just inject into the fat a small amount of the genetic information, this query, this search query that's based on a natural mechanism. All of our bodies, all of our cells can carry out searches. What the company that developed this drug did was they developed a search query, piece of genetic information, that targets a gene that is a master regulator of your cholesterol levels. And human beings who have a mutation in this gene have really, really good, healthy levels of LDL cholesterol. The target gene, if you have a mutation in it, you're healthier than normal. You are less likely to get heart disease and atherosclerosis, you know, the blockage of the blood vessels. So it's a really based on human genetics, and now using a guide RNA that's made in a laboratory, we can inject into any of us. I would be taking this drug if I don't have high cholesterol, but if I did, I would want this drug because I know how it works and I'm not afraid of it. It was one of the safest drugs in this clinical trial that was run by the medicines company that anyone has ever seen. It was in 14,000 people, and the worst reaction was a redness near the site of injection that went away after a few days. And right now, the drug is only approved for people who are failing to control their LDL on statins. But in the future, it will be available to probably all of us in the room, and I would, based on what I know, I would take it because you can't have LDL that's too low. Plus, it's fully reversible, but it lasts for, the newer versions will probably even last longer, but right now it lasts for 3 to 6 months from one dose. So you don't have to take a pill every day, just 2 or 3 injections a year, and you won't, very likely you won't get heart disease. You won't get a heart attack. I think it's a great drug. I think there's a lot more like it that are going to, and I have no financial interest in the company at all, but I would take this drug if it was available to me because I know how it works, and I think it's great because, you know, it doesn't permanently change anything. It blocks the information temporarily, and it's based on very solid information on human genetics. We call it genetics when we compare people who, some are healthy and some are less healthy, some get heart disease, some don't. You can compare people and then figure out what genes are responsible for those differences. And that has led to this drug, and many others actually are now being developed also based on human genetic differences. And that is differences, as you all know, some people live to be 100 years old and they're just still as smart as can be. They haven't lost a single step in terms of their intellectual capabilities. Those people are being sequenced. You know, some of them have some interesting genetics. Some of them have very healthy lifestyles too, but some of them have, you know, there's much that we can learn from that. So this is why a drug like this inclisiran, which is now called Leqvio, is exciting because of the way that it was based on human genetics, on information about people who are healthier than normal. And so it's an opportunity to make medicine, again, based on information. So the same team that developed inclisiran, I was able to recruit them to a company that's now developing a therapy for preeclampsia. This is a condition of pregnancy which leads to the mother having to make a terrible choice to keep the child in her body and risk dying herself, or deliver the baby too early and risk the baby's life. This is a terrible decision, and many women die, many babies die before they, you know, or die in the ICU or have to struggle in the intensive care unit for a long time after birth. And we found the gene that causes this disease. I shouldn't say we, colleagues at Mass General Hospital, Harvard, Anant Karamanchi laboratory discovered a gene that contributes to the cause, actually drives the loss of the syndrome, which leads to high blood pressure and the mother unable to continue the pregnancy. That gene is now a target for this company, and it will go into pregnant women next year. And if it works like it did in the animal trials, it may allow the mother to carry the baby to term from one subcutaneous dose of the drug. So we're crossing our fingers, but that drug has already passed the safety trial in non-pregnant women and will go into pregnant women next year. That's how safe it is. Can you imagine? We had to raise $100 million from really, really smart investors, and they're making a drug that's going to be injected into pregnant women. That's the hardest. There's no one makes drugs for women, and hardly anyone makes drugs for pregnant women. It's like zero drugs, because, you know, and especially an injectable. So this is how safe it is. These drugs are very, very safe because it's a natural mechanism, and, you know, there's a lot of opportunity to, you know, improve and save lives using this. So again, you know, some interesting, very exciting things are coming.
Craig ha detto una cosa fondamentale, cioè la paura. Noi abbiamo sperimentato questa paura durante il Covid, e il risultato di tutta questa informazione, che poi è troppa informazione e disinformazione, è che non si fanno più vaccinazioni contro il morbillo nei bambini in Italia. Allora io mi domando, lo sforzo che Craig chiede a tutti noi e che tutti noi cerchiamo da 25 anni di fare è proprio quello di cercare di raccontare come la scienza funziona. E quando una medicina viene messa sul mercato, ha fatto una serie di sperimentazioni a tutela ovviamente della persona malata, non a favore della compagnia farmaceutica che la produce. È ovvio che ci possono essere dei medicinali che poi vengono ritirati dal mercato dopo che sono messi in commercio, perché si rivelano magari per una certa tipologia di popolazione non efficaci. Ma il sistema, il metodo scientifico, che è semplicemente un metodo che si applica, ed è complicato poter portare una molecola sul mercato, ci vogliono 20, 15, 20 anni, ci vogliono veramente 1 miliardo, 2 miliardi di dollari. Non è una cosa facile, è una scelta anche imprenditoriale importante, non solo legata a chi soffre, ma il metodo scientifico, il modo con cui sperimentiamo le medicine oggi è l'unico che dà una certa garanzia, ovviamente non assoluta, ma una garanzia a tutela della persona che assume farmaci. Se pensate di assumere farmaci che fanno solo bene, pensate male, perché i farmaci hanno effetti ovviamente protettivi, ma anche collaterali, questo si sa. E quindi il medico sceglie in scienza e coscienza tra i rischi e i benefici se somministrarlo, e giustamente la persona malata sceglie di assumere una certa terapia. Ma è chiaro che se l'ambiente che si crea attorno è un ambiente appunto terroristico, in cui si dice che il vaccino anti-Covid, 13 miliardi di somministrazioni nel mondo, è un vaccino poco sicuro perché non è stato sperimentato quando si è iniziati negli anni 60 a sviluppare questi vaccini. Ce lo racconterà Drew Weissman, è chiaro che c'è qualcosa che non torna rispetto alla comunicazione della scienza. E questo è proprio il punto essenziale, perché è ovvio che tutto ciò che non si conosce fa paura, ma la soluzione è cercare di conoscere, non evitare ciò che fa paura, ma conoscerlo per cercare di far sì che faccia meno paura. C'era una domanda qui davanti, in seconda fila, prego signora.
Cosa ne pensi degli studi di espansione del genoma e quindi la creazione di nuove informazioni artificiali?
Sai che non ho capito.
Cosa ne pensa degli studi di espansione del genoma?
Ah,
quindi...
ok, grazie mille, mi scusi.
I'm afraid I'm not sure what you mean by that. Maybe genome editing? Expansion, making a genome larger? I don't know anything about it. So,
date un microfono alla signora che magari ci spiega un pochino meglio. She's rephrasing the question.
E le triplette del genoma che sono più di quelle che codificano per gli amminoacidi.
Oh, okay, I got you now. I know what you're talking about.
Grazie.
Yes, good. That's interesting. There's one of the trends right now is to try to make cells that have a new, a free. I mean, this is kind of technical. It's a good question. Maybe we should talk after, because you might know more about it than I do. But you basically are trying to free up one of the codons in the repertoire of the cell so that you can make a new word. I told you there's 20 words in the language. And there's 20 words in the human, there's 20 words in bacteria, there's 20 words in COVID. Everything alive has just 20 words. And the question, I believe, is referring to the idea of let's make a 21st word. Let's make a new word, because there's room in the genetic code to add an additional word. I mean, it's not a problem. This is why, remember, the kid reading the newspaper, I was like, wow, this is amazing. The language is still the same. Why is the language still the same? Why does the human and the bacterial cell, why do they still speak the same language? Humans now, we're trying to add, some labs have succeeded in adding an additional word. It's really very experimental, but the idea is that you could use that additional word to essentially add functionality, to create a new amino acid. I'd like to hear, I'm skeptical it's going to work, but I think it's an interesting idea, and there might be some applications that would be interesting. I'd like to hear more about it. Are you going to have that, someone speak about that?
Not this year, but we will take this as an opportunity to select, please.
Yeah. Maybe she
Nicola,
can
take
speak
note,
in the
and
future.
next year we will.
Actually, that's something really important. You look pretty young, so if you're not already a scientist, maybe you should become one, because we need more scientists.
Un bel endorsement che ha avuto da Craig. Tornerà. Maybe we can ask her to come back in a couple of years to understand. Maybe she will discover something incredible. Prego.
Grazie. Mi scusi, io non ho una formazione scientifica, non sono una scienziata, quindi le farò una domanda di altra natura. Mentre l'ascoltavo mi è venuto in mente che stamattina per radio ho sentito dire da un professore di letteratura francese che per il poeta francese Baudelaire l'intelligenza umana, l'unica vera caratteristica dell'intelligenza umana era la capacità di immaginare. Ora che ho ascoltato lei, che penso a un bambino di fronte ad una scatola di cereali, penso che forse quello che caratterizza l'intelligenza umana è la capacità di stupirsi di fronte alla meraviglia del mondo. Lei che passa le giornate di fronte a questi enti, esseri così intelligenti, comunicano, trasmettono informazioni, ricordano, traducono, sbagliano, trattengono. Secondo lei cos'è che caratterizza l'intelligenza umana su tutte le altre intelligenze con cui lei ha quotidianamente a che fare? E chiudo, la ringrazio. Io vengo a BergamoScienza perché rimango incantata dallo stupore di persone della sua levatura che non hanno smesso di essere quel bambino, quindi grazie.
No, grazie a lei signora per essere qua e per la domanda che ha fatto, che è molto importante.
Thank you. That is a wonderful question. And I clearly, you understand. It's the curiosity, it's the surprise, it's the wonder, it's the beauty. How could you look at the starry sky at night and not marvel at the beauty of the universe? And I call it the human condition. I think there's that intelligence and humility go together. You have to be humble because it's humbling. It's humbling to see the wonder of the world around you, to open your eyes to the wonder of it and let it just amaze you. That's what's important. That's where I think our sense of purpose in life ultimately comes from. We are born into this universe and we don't know how we got here. Maybe we're told story, we learn a religion, we are sometimes forced, even coerced into believing things, like poor Galileo, who he knew that the earth wasn't the center, and he showed the inquisitors through his telescope. He showed them the moons of Jupiter, which, if you haven't seen them, are so stunning, so beautiful and unexpected. You can see them with binoculars, just with binoculars to your eyes. And he, not only did he discover the moons of Jupiter in his little telescope, he charted their orbits and figured out where they would be in their orbit 100 years in the future, and published it in a book where each line of the book told you at each hour where the moons of Jupiter would be. And you know what he realized? It was a clock. He realized that the moons of Jupiter gave him a clock that would show the same time everywhere in the world. And that's why he published it. Captains took his chart, his notebook of timetables of the planet, of the moons of Jupiter. You can see them through your little binoculars and through a little telescope from a deck of a boat. They could look at the moons of Jupiter and they could tell what time it was exactly. So they would actually be able to navigate accurately using that as their clock. So I'm just giving that as an example, but just imagine the wonder of the people who made the first, who first got those images from the space telescope, where they exposed the camera first it was the Hubble and now it's the Webb telescope that's even better. But they chose a part of the sky where there are no stars. There are no stars in the galaxy, in our galaxy, and they just made a really, really long exposure. And after like 7 days of exposure, they developed this image showing that black space in the sky is full of galaxies. It's just incredible, right? We're this tiny speck. This is where the humility comes, you know? And there's certainly room for God. I'm not an atheist. I consider myself spiritual because I'm awed by the beauty, by the wonder of our existence. And I admit I do not know where I came from. I don't know. And I'm not going to try to tell you a story because I don't know. You know, you maybe want to tell me, you know, God made the universe and, you know, created humans and gave us a soul. I don't know. I don't know where we came from. I don't know where God came from. And I'm okay with that. And I think that's the human condition. And it goes with a sense of wonder. You know, I feel like if you're afraid, if you fear, then you lose so much. You know, let the fear go and just look up at the night sky and just wonder. I think there's a beauty there. And I think that's where a lot of science, I think that's where art and great literature, I think it all comes from that same spark of wonder.
È un tema molto affascinante, ovviamente. Altre domande dal pubblico? Se no riprendiamo con Agnese, se non c'è nessun altro che ha voglia di interloquire con Craig. Ce n'è una? No, stai parlando di no. Eccola là, vedi che l'abbiamo sollecitata. Prego ragazzi.
Io volevo fare una domanda in realtà piuttosto semplice, nel senso, tutti noi da bambini abbiamo dei sogni e vincere un premio Nobel della medicina penso che sia un grande sogno da realizzare.
L'incubo più che un sogno. È quello che bisogna fare.
È un grande sogno che lei ha realizzato. Io volevo capire quando il bambino che leggeva le scatole di cereali ha effettivamente capito di avere scoperto qualcosa che avrebbe poi cambiato quello che è il corso della medicina.
Bella domanda.
Well, I made the decision when I started working to work on a tiny microscopic animal. It's actually a little worm. And I chose it in part because I guess I understood that humans and worms are actually very similar to each other. So, but I certainly never expected, I really never expected that what we would discover would lead to a new medicine. I thought maybe we would discover something just fundamental about how organisms handle information. And so, yeah, I was incredibly surprised when we found that the organism had this incredible response to the RNA that we were injecting into them. My fascination with information led me to want to put information back into an animal. And obviously, you remember in the newspaper article, the human gene was put into the bacterial cell and it could work. I wanted to put human genes back into humans. You know, like if you have a mutation that's making you sick, maybe we can fix it. But I was really, more interested in the fundamental mechanism. I was curious about that. And what we discovered quite accidentally is that when we put information in the form of RNA into a the animal responded in a remarkable way. The RNA could essentially turn a gene off, you know, in this little tiny animal. And it did it so specifically, so perfectly, that the genetic information in the RNA that we were giving the animal matched perfectly with the gene that was getting turned off. And we then set about to try to find the mechanism, because it was so unexpected, really, truly unexpected. These animals could respond, you know, perfectly to the query that we were giving them. We could even feed them the RNA, and they would turn off the gene that we targeted. So we were really excited and wondered what could possibly be going on. And so we did what's called a genetic screen, a mutant hunt. We hunted for the genes that the animal needs in order to carry out the search. So there's a way, when you're working with a tiny little organism like this that grows, just takes 3 days for the animal to reproduce and make 300 progeny, so you can grow a lot of them. We could treat them with a mutagen that damages their DNA, and then search for mutants that are resistant to the RNA that we target them with. So we target them with an RNA that silences a gene, and we look for mutants that can't silence the gene anymore. They're mutant. They don't do it anymore. And then we found out what the broken part was. We cloned the gene and found out what it encodes. It encodes a protein that humans have. So the worms have it, and the humans have it. The same protein. In fact, plants have it, even some bacteria have it. So the protein is encoded by a messenger RNA that the worm makes. The human makes the same protein. And the human protein and the worm protein both get loaded with guide RNAs. And it's really cool what they do. They hold on to the end. This is a polymer, and it's only 20 nucleotides, only 20 letters long, but they grab one end of that guide, and they hold it tightly, and then they present the information in the guide to targets. And do you know how fast they do it? They do it at 100 million collisions per second. That's the diffusion limit, it's called. It's how fast living things move inside the cell. It's called Brownian motion. These guides are bouncing and bumping into targets, and if they match, then they can do this, Watson-Crick staircase, this helical turn that so precisely maps the information in one of the strands of the helix to the information on the other strand. If they match perfectly, it builds a stair in the staircase. And they can do that at millions of collisions per second. They carry out this search, and they find the matching information, and boom, when they form if the guide RNA wraps the target perfectly, makes perfect staircase steps, then the enzyme, the protein that's holding, can cleave the target strand, cutting the information and destroying the information that it found. So it can search, it can find, and it can silence its target. And worms had it, and this was 1999 when we described the first gene. So a year after our paper, 1999, we found the first gene, and we found out that humans have it. And that was exciting because now it wasn't just a worm thing. It was potentially something that would be useful not only for humans, but also for animal and plants. There are genetic tools. You can do this for crop plants. You can genetically modify them to make them drought resistant or resistant to to viruses or microbes or et cetera. So there's a lot of opportunities. And to make a long story short, it was just incredible to discover you know, something so fundamental. You know, this was before the company Google went public. I wish, you know, in 1999, I wish I had bought stock in Google, because I still was trying to figure out, well, what is this thing we discovered? You know, it's guided search, but when I grew up, you had to look in the index in the back of the book and find the word. You know, there was no guided search. Now we understand you can't live without guided search. And all organisms have it. Everything on the planet uses guided search. And this relates to also this CRISPR thing I was asking about earlier, because CRISPR is guided search, which enables genome editing, because you can modify DNA with this particular type of enzyme. Anyway, long answer, sorry. But there's still, you know, there's still many, many things still to discover, and really exciting, completely, I'm sure, very surprising discoveries in biology yet to be made. So I'm still at it. I love it. I'll never stop. It's just, it's fun, and there's so much still to learn.
Allora, abbiamo fatto le 10:30, 2 ore che stiamo conversando amorevolmente, sono tante. Allora, se c'è un'ultima domanda veloce, rapida, che volete fare a Craig, in maniera tale che questa conversazione veramente possa soddisfare tutti quelli che hanno partecipato. Se non c'è, faccio io un'ultima domanda, se Agnese e Nicola mi permettono di farla. Vorrei tornare sul concetto della vita. Il modello gerarchico della vita, come tu ben sai, prevede che le informazioni vadano solo in maniera ascendente, ovvero sia dall'atomo alla molecola all'organismo. Il problema serio, e quindi da qua nasce il concetto di riduzionismo tipico, insomma, dello scienziato, di un certo tipo di scienziati. Il problema è che tu hai fatto una scelta, una scoperta che ha cambiato radicalmente, ha messo in crisi questo modello, perché noi adesso sappiamo che tramite l'epigenetica cosiddetta, ovvero sia tutti quei regolatori, diciamo così, del messaggio codificato dal DNA, è possibile che anche l'ambiente, in qualche modo, in maniera discendente, possa modificare gli aspetti biologici. Ci sono una serie di scoperte importanti. La prima di tutte fu quella che fu dimostrato in maniera molto semplice, che una topolina che non accudiva i propri piccoli rendeva questi molto più stressati, perché non veniva espresso un recettore, quello dei cosiddetti glucocorticoidi o cortisolo, o insomma ormoni dello stress, nell'ippocampo, che è una piccola area del cervello. Quindi fisicamente il non accudire, il non coccolare, il non toccare, il non allattare i propri piccoli portava a questa alterazione biologica, come se a quel punto fosse in qualche modo, diciamo, l'informazione andasse al contrario, no? Fosse discendente. Noi sappiamo che bambini maltrattati o accuditi male purtroppo sviluppano molte più malattie psichiatriche e psicologiche quando sono adulti, lo sappiamo da tempo, però questo ci ha dato la dimostrazione biologica che anche le malattie psichiatriche, proprio perché possono esistere delle cause discendenti, possono avere, tutto sommato, possono in qualche modo essere o dipendere anche di quella che è stata la storia del singolo individuo e non la storia del suo genoma. E questa, diciamo, posizione si chiama emergentismo, una posizione che nasce a fine Ottocento in Inghilterra, insomma, poi si sviluppa, poi è passata un po' di moda quando è stato scoperto il DNA, perché ovviamente tutti pensavano di aver risolto tutto, bastava leggerlo. Poi sei arrivato tu e hai detto, non è proprio così, non basta leggerlo, non basta tradurlo, c'è tutto un sistema di regolazione che è importante. Ecco, quindi la mia domanda è, tu accetti dal punto di vista teorico che ci possono essere delle cause discendenti,
cioè che l'ambiente possa modificare biologicamente, ovviamente non la struttura del DNA, ma la propria funzione? Cioè credi che le evidenze scientifiche che abbiamo sono sufficienti oggi a sostenere che in un evento biologico le cause possono essere sia ascendenti sia discendenti? Spero di essermi spiegato, Craig.
Thank you. You promised a short question. That was not short, but that's okay. One of the things I love in biology is something which we sometimes call emergent property. It's something that happens, you know, probably unintentionally, but it's you know, but it has real importance. So, for example, an emergent property of human intelligence, right? We develop our abilities because it's really useful. Our mental, you know, our brain, our intelligence, if you will, evolves because it's useful. It helps us predict the future. We can predict the future better, we're more likely to survive, right? Well, what's one of the unfortunate consequences of being good at predicting the future, right? You realize your own mortality. You know, sadly, you know, we got ourselves in trouble by being able to predict the future, and then we start asking all the questions, you know, that come with that, like, where did we come from? How did I get here? You know, and for millions of years, humans, pretty much every bit as smart as us, lived on this planet, but had no spoken, well, probably they had some spoken, but no written languages. So every time a culture died, all the knowledge died. When the culture died, the language died. And that happened over and over again in human history until somebody figured out how to write stuff down and save, you know, the culture from generation to generation. It's really fascinating to think about how difficult the emergence of human sort of culture has been. And yet now culture is really the fundamental way, that the thing that makes us human is our ability to share that, evolve culturally rather than biologically. We can adapt now using our intelligence to plan for the future. It's really beautiful, but it's also created lots of problems. Living things, I think, never gave up on the important information from the environment. The environment essentially is part of us. You know, the earth gave rise to life, we think. And the information from the earth, you know, was captured, the sunlight, the energy, you know, molecules doing chemistry essentially were influenced by their environment, and the building blocks began to assemble, and that never stopped. It was a fire that started billions of years ago. It was an informational fire that started on this planet. And the molecules that could copy themselves better made more copies of themselves and persisted. And they could eat the other molecules, you know, they could harvest the energy from their environment. And that's the beginning. But there, these layers of regulation, you know, the DNA became important later, probably. The informational molecules that most likely gave rise to life were much like what we now call RNA. RNA is more reactive than DNA. DNA is more inert. It can't catalyze its own cleavage as easily, but RNA can easily excise itself. And there are viruses, they call them, well, it's not important, it's just jargon, but they're viruses that basically cut themselves out and then precisely insert themselves somewhere else in a message. They can cut themselves out of an RNA. And, you know, they're just living, life is just full of these surprises if you start looking. And I think there's a lot still to learn about how information is stored in you know, and how the environment influences information. They used to call information that was not encoded, you know, Lamarckian, or, you know, something, you know, like that could be influenced externally that became part of the program. And there are ways now, we understand how organisms handle information. We can actually see how a gene could be selected, you know, based on an environmental change. A gene could be turned off, for example, by a change in condition, and that would be propagated. So we can see these types of regulations. So to answer your long question, I could go on, but I think there are a lot of surprises still to come in biology, and a lot of them have to do with this question of how is the information propagated? Where is the robustness? How do you get, we are so complicated. You know, our biology, it's incredible what we do. I mean, nobody asked about how our body makes, you know, antibodies. You know, how do we fight diseases? But it's like, it's truly incredible. I mean, you should read about it. If you haven't, you can read about how your body reacts to something like the mRNA vaccine. How do you actually get immunity to anything? Read about it. It's in the textbooks. You can read about it. It's like, they don't say, this is incredible, this is surprising, but every other word should be, this is incredible, this is surprising. Because just in a nutshell, I'll tell you what our body does. Our body presents every single different protein that you and I can make. It makes them in a special organ called the thymus. And then if an antibody reacts to a protein that's part of us, the cell that expresses that antibody dies. It's called educating our immune system. Our immune system learns to not attack our own body, but it randomly it has the ability to react to anything, but it teaches itself not to react to our own body. That's incredible. You know, the textbook chapter should start with, it's incredible how the immune system works, because it's incredible. It really is. I mean, it's truly incredible. Our body learns, it makes antibodies randomly to everything, and then it teaches itself which things to not express, because you don't want to kill yourself, right? That's the fundamental rule of biology, is if you kill yourself, you die and you're gone, and that's it. You're not going to be around next generation. But our bodies are amazing, and our immune system, our antibody immune system, is just something that you can look up and you will go, oh my, that's incredible. That is truly incredible how we do it. We can make trillions of different antibodies by recombining our own DNA. So you start out when you're born with a genome that has, you know, no antibody diversity, and it can assemble an incredible diversity. And all the COVID vaccine does is it gives our body a heads up, hey, if you can make an antibody that sticks to this, then you should do it, because this thing is in you now. It's not good, and you better be ready to destroy it, right, or get rid of it. That's how you get immunity. Your body has to learn. It has to select the antibody forming cells that react to this new antigen. And at the same time, they don't tell you this all the time, your body has to learn not to react to yourself. It has to learn. It's like, it's pretty incredible. And I challenge you, if you're not a biologist, look it up. Learn a little bit about how your body protects itself. and then think about the vaccine and what it means to get vaccinated. And you'll feel a lot more comfortable because our bodies are way, way, way more incredible and sophisticated than we give them credit for, right? I mean, we are just beginning to learn and understand how we work, and we are incredible. So, anyway, I should stop.
Questa è una conclusione, diciamo, fondamentale, perché quello che sta dicendo Craig è quello a cui assisteremo nei prossimi probabilmente 50-100 anni, cioè stiamo capendo come funzioniamo, stiamo capendo che siamo riusciti a resistere per 3 miliardi e mezzo continuando ad evolverci, abbiamo capito che abbiamo sviluppato internamente dei modelli di difesa e adesso non facciamo altro che copiarli. Adesso stiamo usando questi anticorpi nelle terapie, addirittura terapie nell'oncologia, nelle malattie autoimmuni, stiamo usando le cellule per sostituire cellule danneggiate, usiamo i regolatori della trascrizione genica che ha scoperto Craig per fare i vaccini o per fare appunto delle molecole, cioè usiamo queste terapie, per definizione sono quelle che a me piace chiamare la vera medicina naturale. È ovvio che è probabile che abbiano meno effetti collaterali perché sono semplicemente un potenziamento delle difese naturali che ci hanno portato a vivere fino a 80 e passa anni in pochi secoli di vita e ci hanno permesso di essere comunque una specie sana da un certo punto di vista. Quindi questa è la speranza per il futuro, di avere farmaci sempre più potenti, sempre meno tossici, sempre più efficaci, che copiano quello che la natura ci ha insegnato e copiandolo probabilmente riusciranno anche a curarci meglio. Io chiudo qua la serata, è stata molto bella, vi ringrazio veramente tutti per aver partecipato in questa conversazione, Craig in primis. E vi aspettiamo tutti domani alle 5 in aula Magna Sant'Agostino per l'inaugurazione. Craig, the podium is yours. Grazie a tutti.
And thank you to the translators. Thank you.